Hi, my name is Allison Pennell Townsend. I am going to be talking about the general human cell and its organelles. I have a diagram here of a cell. I've mostly used pasta noodles to represent a number of organelles. We'll start with the nucleus, which is the largest organelle. It contains the cell's chromosomes. So it is the genetic control center. It directs protein synthesis. It contains the nucleolus that produces ribosomes. Next, you see the rough endoplasmic reticulum. It's called a little network of cytoplasm. It's a system, or the endoplasmic reticulum altogether is called the little network within the cytoplasm. It's a system of interconnected channels called cisterns. Specifically, the rough ER produce and synthesize proteins. They have the appearance of fingerprints. The hallways tend to be parallel and make broad arching curves, and they do have the ribosomes. The smooth ER, which I have used macaroni noodles to represent, are abundant in cells that do a lot of detoxification, such as the liver and kidney cells, and in cells that synthesize steroid hormones. They also are storage for calcium and lipid synthesis. And as you can see, they do not have ribosomes, and they are more tubular and branch more extensively. Going back to the ribosomes, that will be the next organelle I'll speak about. Ribosomes are small granules of protein and RNA. They interpret the genetic code. They synthesize polypeptides. They are produced in the nucleolus of the nucleus, and they're found attached to the rough endoplasmic reticulum, nuclei, nucleoli, and in the cytosol. Next represented, I have the Golgi complex. Again, I've used fettuccine noodles to represent the Golgi complex. They tend to have rounded edges. They have small stacks of the cisternae. The Golgi complex synthesizes carbohydrates and puts the finishing touches on protein and peptide synthesis. It packages cell products into Golgi vesicles. I have Golgi vesicles here below. The Golgi vesicles sometimes export products out through the cell membranes through exocytosis. And it's the most mature cisternae that break up into these Golgi vesicles. And some of these Golgi vesicles become lysosomes. And I have lysosomes next. Lysosomes are a package of enzymes bounded by a membrane. It's a specialized type of Golgi vesicle. It contains digestive enzymes. It travels through the cell and breaks down unwanted materials. And it's oftentimes darker than the peroxisomes. And so the peroxisomes are going to be what I speak of next. The peroxisomes are made by the mitochondria, or the mitochondrion for singular. The general function of the peroxisome is to use molecular oxygen to oxidize organic molecules. It helps detoxify the cystosol by producing hydrogen peroxide to break down particles. The detoxification of free radicals, alcohol, and other drugs. It oxidizes fatty acids. And as I said before, it's produced by the endoplasmic reticulum and the mitochondria. It does not have connections with the Golgi complex. The peroxisome is abundant in the liver and kidney cells. So next I have the mitochondria, and they have a system of cisternae, just like some of the other organelles do. The mitochondria is called the powerhouse of the cell. It synthesizes ATP. Energy is not made here, but it is extracted from organic compounds and transferred to ATP. They sometimes undergo fusion and fission. It has a double membrane. They're quite mobile. It has internal folds known as cristae. The space between the cristae, the matrix, has ribosomes, as you see here, enzymes, and DNA molecules. The next organelle that I will speak of is the centriole. I am not an artist, cannot find food to represent the centriole, so here is my drawing. The centrioles, they're paired organelles. There's 9 groups of 3. They look like a pinwheel. They're used in cell division. During cell division, microtubules are responsible for moving and arranging the components within the cytosol to ensure the cell divides equally. And here I have indicated the centrosome, which is a clear patch of cytoplasm near the nucleus containing a pair of centrioles. So I'm going to do one more overview of my diagram. I do have the microvillus represented. I do have the secretory vesicle. I do have a microtubule represented by a spaghetti noodle. I do have the plasma membrane, the inner and outer represented here again with spaghetti noodles. Thank you.